Hello everyone, how are you? Welcome to this amazing platform of Infinity Learn by Sri Chaitanya. My lovely students, my name is Kiran and I am your biology faculty. Let us talk about today's session, students. In this class, we will continue our discussion on transcription process. Remember, in the previous class, we were studying about or discussing together how transcription process get completed or takes place in a prokaryotic cell like a bacteria, right. So, that was the objective of previous class. In this class, we will try to understand how process of transcription. What is transcription? Copying of genetic information from one of the strand of DNA into an RNA molecule, and not to forget, not the entire length of the DNA, rather a small segment of DNA will participate, and from that only one strand will participate in transcription process, that is copying genetic information from DNA to RNA. So, that is a quick recap of a transcription process. Now, let us understand in this session how this transcription process, that means formation of RNA molecules, takes place inside an eukaryotic cell. Students, before starting the session, I would love to tell you that please do visit infinitylearn.com website and check out the courses that we offer there. You can use those courses and subscription at 40% off by applying the code Kiran40. So, I request you all, go check out the courses and do not forget to use the code Kiran40 to get straight 40% off on the various subscription and courses that we offer at our platform infinitylearn.com website, right. Now, let us start the session, students. See students, what we need to understand in this is, first of all, let us understand what is the place in an eukaryotic cell. See, transcription process we are studying in a eukaryotes, right, in a eukaryotic cell. So, let us talk about the place first. Remember, when I was teaching you transcription process in a prokaryotic cell, at that time I told you the place will be cytoplasm, because there are no well defined or membrane bound organelles inside a prokaryotic cell. Hence, transcription process was taking place inside cytoplasm, but this is not the case in case of eukaryotic cells. Eukaryotic cells possess well defined membrane bound organelles, nucleus, etc., right. Hence, please mark the position where the entire process of transcription in a eukaryotic cell will take place, inside nucleus. Please remember this very important fact. Now, students, let us compare the process of transcription in a prokaryotic cell and a eukaryotic cell before I start explaining how this process of transcription takes place inside an eukaryotic cell. So, listen this very carefully. There are two additional complexities. I repeat myself, there are two additional complexities that are there during the process, or that comes during the process of transcription when it takes place in a eukaryotic cell. What are these two complexities? The first one is presence of at least 3 types of RNA polymerase other than the one that are present in the cell organelles, right, where RNA formation of RNA takes place other than nucleus, right. So, presence of at least 3 types of RNA polymerase itself complicates the process of transcription in eukaryotes. We will definitely check out what are these various 3 types of RNA polymerase and exactly what type of RNA molecules they will produce, right, individually. That is the second objective, right. Now, second complexity. What is the second complexity when it comes to transcription process in a eukaryotic cell? The process of splicing. Yes, students, do not forget that when a primary transcript, what is a primary transcript? A newly synthesized RNA molecule. I repeat myself, when a primary transcript is formed during transcription process in eukaryotic cell, that means a newly synthesized RNA molecules actually possess introns also along with exons. Yes, and the presence of introns in a primary transcript of RNA polymerase, the newly synthesized RNA polymerase need to be processed, right. So, this is the second additional complexity in a transcription process when it comes to eukaryotic cell. So, during the processing of a primary transcript RNA, what will happen? The interons, what are interons? The non-coding sequences, right. So, the interons will be removed from the exons part and the remaining exons will be added, right, in a specific manner. So, these are the two important complexities that takes place when it comes to transcription process with respect to eukaryotic cell. Presence of at least 3 types of RNA polymerase and second processing of the primary transcript. So, this is the difference between when it comes to transcription taking place inside a bacterial or a prokaryotic cell and a eukaryotic cell. Now, let us take both these complexities of transcription process with respect to eukaryotic cells in detail. So, we will start with the very first one, this presence of more than at least 3 types of RNA polymerase, and let us see what are the different types of RNA that these respective RNA polymerase will synthesize. So, see, at least 3 types of RNA polymerase, there are 3 types, RNA polymerase 1, 2 and 3, and notice students, I want everybody to notice first on the second one. So, RNA polymerase 2 will actually synthesize hnRNA. What is hnRNA? It is a heterogeneous nuclear RNA. hnRNA is a precursor, right, a precursor of mRNA. That means RNA polymerase 2 will form hnRNA, which is a heterogeneous nuclear RNA, and this hnRNA is actually the precursor of mRNA. That means this hnRNA, which is a primary transcript, a newly synthesized RNA molecule, when this hnRNA will undergo processing, after the completion of processing, it will produce what? It will produce a mRNA, right. This mRNA will participate in the translation process for the formation of polypeptide chain. So, please notice students, RNA polymerase 2 will synthesize a primary transcript hnRNA that is actually a precursor of mRNA. After processing gets completed in this hnRNA, there will be formation of an mRNA that can participate in translation process, or that possess the codes for coding that will decide the type and the sequence of amino acid that should be there in the protein that will be coded from that particular mRNA. I hope this makes proper sense and you are able to understand what I am trying to convey. Second, let us talk about RNA polymerase 1 and RNA polymerase 3 as well. So, RNA polymerase 1 will produce 5.8, 18S and 28S rRNA. Please note this point. Talking about RNA polymerase 3, it will form tRNA. Remember tRNA? Adapter RNA. This tRNA will actually read the codes on the mRNA with the help of anticodon loop and bring the respective amino acid during the process of translation. So, it is the tRNA that is going to read the codons. Do not forget, very important for the translation process, right? And RNA polymerase 3, other than producing tRNA, will produce 5S rRNA and snRNA. What are these snRNA? These are small nuclear RNA, and what is the function of these snRNAs? Not to forget students, here if I talk about the varieties of RNA, you can majorly see 3 types of RNA. hnRNA, which is going to produce mRNA after processing, and what is the function of mRNA? It possesses or it holds the sequence, right? It possesses the codons, it possesses the codes according to which amino acids will be arranged and protein will be formed, right? It is going to act as a template. Next is the rRNA, various types of rRNA. Do not forget students, rRNA, the main function of rRNA is the structural one. They will help in the formation of ribosomes, right? And next that you need to understand is the another diversity. If I talk about RNA molecules that are produced here, the another type is the tRNA that is going to act as an adapter molecule. I have already done the explanation itself that what is the function it is going to play during the translation process. Now, last type that I want to discuss about the type of RNAs that are going to be produced from these respective RNA polymerase is snRNA. Let us discuss in little detail what this snRNA is going to do. So, this snRNA or small nuclear RNA will actually help in formation of spliceosomes. And what are spliceosomes, students? Spliceosomes is a complex that will help during the splicing process. What is a splicing process? Splicing process is a process during which from a primary transcript that possess introns and exons. Remember, exons are the one that get coded and introns are the one that do not get coded. So, spliceosomes which will be formed. Who is going to help in formation of spliceosomes? So, snRNA along with certain proteins will help in formation of a complex known as spliceosome, and this spliceosome will help in the splicing process during which from a primary transcript of RNA introns will be removed, right? This is the function of snRNA. And my kids, please notice, if you notice these three types of RNA polymerase and the types of RNA they are making, it clearly represents what? A clear-cut division of labor or work. So, all types of RNA molecules, unlike in a prokaryotic cell or a bacterial cell where all types of RNA molecules were being, almost all types of RNA molecules were being produced or synthesized with the help of single RNA polymerase. That is not the scene when it comes to eukaryotic cell. In eukaryotic cells, several RNA polymerase participates to produce different types of RNA molecules. So, there is a clear-cut division of labor. I hope this much is clear to all of you. Let us see, students, now next process. So, see, students, if you observe this, what is this? This is how transcription process is going to take place. So, in your NCERT, transcription, during the transcription process or where the explanation of transcription process is given, there majorly they are talking about the formation or synthesis of mRNA. So, mRNA is actually taken into the center in order to, or as an example, in order to explain the transcription process in eukaryotic cells. So, we will also keep this thing or this discussion in center. So, let us see how mRNA will be synthesized during transcription process in eukaryotic cell. So, see, students, during the transcription process in eukaryotic cell, first of all, who will be produced? A primary transcript will be produced, right? What is this? This is a primary transcript. What is this, my kids? This is a primary transcript. What is a primary transcript? If I talk about in words, it is a newly synthesized RNA. So, during the transcription process, first of all, there will be a formation of a primary transcript. What is a primary transcript? It is a newly synthesized RNA molecule. Now, this primary transcript will actually possess introns as well as exons, right? So, it contains both introns and exon, and hence it is non-functional. Why? Because if it directly participates in the process of translation, what will happen? This entire RNA molecule, primary transcript RNA molecule will possess or will have interrupted coding sequences that will actually complicate the process or mechanism of translation, that means formation of proteins. So, while formation of protein, what do we need? We need actually a clear cut, like a simple and an obvious molecule of RNA which possess only coding sequences. We do not want the intervening non-coding sequences in a finally matured mRNA molecules, because during the translation process we want this process to be smooth. The process of formation of polypeptide chain should not be complex because of the presence of introns who are actually not going to be coded during the translation process, and hence the primary transcripts are non-functional because of the presence of introns. So, please note students, primary transcripts are non-functional. Second, in order to make them functional, what should be done? The introns should be removed. So, remember, primary transcript will possess both introns and exons. They will possess both introns and exon. And if you notice in this diagram of NCERT, orange portion is represented by introns, the non-coding sequences. Introns are non-coding sequences, and if you notice exon, what are these? These are the coding sequences. These are the coding sequences. So, see kids, right? So, presence of intron and exons in a primary transcript makes it non-functional, and in order to convert it into a functional mRNA, you need to remove the introns from the exons part, right? And that's the exact process known as splicing, in which spliceosomes will help. Remember, snRNA is going to help in making that complexes. Spliceosomes during splicing process will actually remove the introns, the non-coding sequences from the exons, right? So, this primary transcript will be subjected to splicing, where introns are removed from the exons and then remaining exons will be joined in a definite order. Let us see the next thing, students. So, there are two additional what is this hnRNA? This is the primary transcript, this is the hnRNA. This is the hnRNA. Who is the primary transcript? Also known as hnRNA, right? So, this primary transcript or hnRNA will undergo two additional processing. First I want to talk about capping, and then I want to talk about tailing. So, other than splicing, other than removing introns from the exons from hnRNA or primary transcript, which is a precursor of mRNA, two additional processing will take place. There will be addition of cap on the 5' end of the mRNA, and there will be tailing also. That means there will be an addition of a tail on the 3' end as well. So, when there will be a formation of a fully mature mRNA, right, a processed molecule of mRNA, see this, a messenger RNA, mRNA which you are obtaining at the end, there will be a presence of what? There will be a presence of cap and tail, right. So, remember students, these are the two additional processing that will take place. So, what is a cap? Let us talk about the position where it will be placed, and let us also talk about the biochemical nature. So, cap is going to be made up of methyl guanosine triphosphate, and remember, it will be attached to the 5' end of the molecule. Next, tailing. What will happen during tailing? First, tail will be added at the 3' end, and there will be a poly A, right. A poly A means adenylate residue approximately 200 to 300 A. Poly A tail will be added. What is poly A? It is adenylate residue will be added at the 3' end. And then after mRNA is fully processed hnRNA and is transported out of the nucleus for translation, right. So, a fully processed hnRNA, what is a fully processed hnRNA? In which splicing has taken place, in which capping has taken place, in which tailing has taken place. So, after all these processing steps, a fully matured hnRNA will be known as a fully processed hnRNA will be known as mRNA, and this mRNA is ready to be transported outside the nucleus for translation process, which will take place in cytoplasm. Tell me students, is this much clear? Is this much clear? So, observe this part. this is this hnRNA primary transcript, and these are the exons and these are the introns. So during RNA splicing, what are these kids? These are the spliceosomes that will help to remove the introns. So what is happening here? Splicing is happening, because of which, with the help of spliceosomes, introns will be removed, and finally what you will get is a smooth mRNA which will possess only the exons, that is the coding sequences. Next students, two very important thing that I want to discuss with all of you is, yes Killer Jack, hello, good evening. Introns, ma'am, please explain the second point. What was the second point that I could not explain? Capping. Do you want to ask me about capping? Please let me know your doubt clearly in the chat box, then I will come to that point again. Please let me know. I am not understanding which second point you are talking about, Jack. See students, the split gene arrangement. What is a split gene arrangement? Presence of introns and exons. That means introns are the interrupted sequence. Who is interrupting the coding sequences? Exons. It is the presence of introns. So presence of introns and exons in a gene is known as split gene arrangement, and such split gene arrangement is actually the mark of ancient character. So split gene arrangement represent probably an ancient feature of the genome. The presence of intron is the reminiscent of antiquity, and the process of splicing represents the dominance of rRNA wall, right? Because see students, in order to produce a protein, in order to produce a polypeptide chain, what is happening? In order to synthesize a polypeptide chain from a DNA, RNA need to be synthesized. In that RNA also splicing needs to take place, right? So in order to synthesize a protein, a polypeptide chain, DNA is dependent on RNA, and further also many complexities or processes that takes place around the RNA molecule, and hence the world is what? This all processes while doing transcription, while doing translation. If you notice, there is a presence, there is a dominance of RNA molecule, and hence all this represent what's the dominance of RNA word, right? Next, in the recent time, the understanding of RNA and RNA dependent processes like splicing, like transcription, like translation, what are these? These are RNA dependent processes in the living system have assumed more importance. So that's all I wanted to explain in today's session, students. Let me know. Okay, so Jack wants to know about the capping part. Okay Jack, see here, what I told you, hnRNA other than splicing will undergo two additional processing. One is capping and another is tailing, right? So cap will be added on the 5' end of the hnRNA, and tail will be added to the 3' end of the hnRNA, and after this splicing, capping, tailing, this fully processed hnRNA will be converted or will be known as an mRNA, which is ready to be transported out of the nucleus and can participate in translation process. Well, talking about capping, cap is going to be added at 5', it should be clear. Now let us talk about the biochemical nature. What is this cap? Is that a cap that you wear? No, it's cap means there is something, it is taking place where inside a living system. So if I say cap, that means there has to be some chemical composition of this cap which is getting attached with the help of some kind of bond on the 5' end of this primary transcript. So let us talk about the chemical nature. What is the chemical nature? It is methyl guanosine, methylated guanosine triphosphate molecule. Now let us talk about the tail. Tail is a poly A tail, right? Is in an adenylate residue ranging from 200 to 300 attached at the 3' end of the transcript. Tell me Jack, is that okay? Can we conclude the session now Jack? If that is okay, please give me a confirmation Jack. I hope that your doubt is solved. Let me know if it is not. Okay students, that's all I wanted to explain in this session. Thank you for joining my class. Give me a thumbs up if you like me and this session, and do not forget to subscribe Infinity Learn by Sri Chaitanya, and also do not forget to hit the bell icon, so whenever I will come live, you will be notified every time and won't miss any single session in future. That's all students. Bye-bye.